Hello and welcome back to Bio 181. In this lecture, we will discuss genes and their expression. In other words, how they are transcribed into proteins. We've discussed a lot about genetics since the first chapter. We've discussed how DNA is the source of heritable traits, and those traits are recorded as genes. In this chapter, we'll see how those genes lead to the production of proteins that govern the cell's structure, function, and behavior. A gene is a segment of DNA containing a set of instructions for a particular process or product. And a trait is an inherited characteristic of an organism, either physical or behavioral. So, how do we get from genes to traits? Let's start from a high level view and work our way down to the more specifics. Our set of genes are stored in the nucleus, which houses chromosomes that are made up of DNA. This DNA is a polymer of nucleotides. Different sections of the chromosome have nucleotide segments that encode for genes, like different recipes in a recipe book. And variations of the same gene is called an allele. And here again, we can see that on this particular location, chromosome 4, Q arm, and the loci or location 22.1, we find the gene called SNCA. This segment of nucleotides carries the code for the protein alpha synuclein, which is important for the function of the nervous system. And certain alleles, or the same gene with different mutations, can lead to a disease called Parkinson's disease. Nowadays, we have genetic databases that we can rely on to find these genes. This is the NCBI website for the National Institute of Health. And you can look up just about any gene that has been sequenced out there on this website. So here we're looking at our particular alpha-synuclein gene. It's in Homo sapiens species chromosome 4. Here is the selected region where we find it on chromosome 4, being that this is the nucleotide sequence is found on roughly the 89th millionth nucleotide sequence is where we find it on. It's quite a long piece of gene here from top to bottom to encode for this one protein. As you know, DNA is a double helix, and the sequence of one strand is complementary to the sequence of the other, with adenines always pairing up with thymines and guanines always pairing up with cytosines. This fact allows the cell to make copies of the strands by using the other strand as a template. Consequently, when we write out the genetic code, as I showed in the previous example, we do not bother writing out the entire sequence of both strands. Instead, we write a single strand out. Just as you were required to do in the last chapter, the conventional method is to write one of the strands out, starting with the first 5' nucleotide to the last 3' nucleotide. And that's exactly what you'll see here. This is the very beginning, the 5' end of the template strand, all the way to the end. What we do know is that when something goes wrong with alpha-synuclein, the result is Parkinson's disease, or some sort of neurodegenerative disorder. Here is a tiny fraction of such a code written out for alpha-synuclein. In the sequence for a normal or good recipe for making alpha-synuclein, the 209th nucleotide is a guanine, seen highlighted here in a red G. Some people have a different version of the gene, an allele wherein the 209th nucleotide is not guanine, but rather adenine. This example gives you a sense of how important the order of nucleotides in a gene is. And abnormally structured alpha synuclein leads to the formation of what's called Lewy bodies, which are buildups of the alpha synuclein in the neurons of the brain. So, we've seen how we read DNA, but how do the cells do so in order to synthesize proteins? We've seen cells do so in order to make new DNA strands, and there are similarities, but some special differences. It's essential to understand that DNA is very precious. Imagine all the information your cells need to survive is stored within the nucleus. The cells don't want to just take the DNA out of the nucleus to take to the protein factory. Rather, they keep the DNA there and make copies of scripts of the blueprint to send off to the ribosomes. That way, the DNA stays safely in the nucleus. And not to mention, it would be rather complicated mess to have everything working in one single location. So the cell can only access the information in genes by making copies of them. Unlike the original DNA, these copies can be removed from the nucleus for use. Start to finish, when a cell wants to make a particular protein, we can break this down into 3 steps. First, the sequence of DNA code for a gene is copied into a molecule of RNA. This is called transcription because it is like you taking notes or transcribing. The second step is editing of the RNA sequence so that it is properly prepared for use. This is called post-transcriptional modification. Lastly, the RNA sequence is read and converted into a protein sequence. In a way, we've changed languages from nucleic acids in the RNA polymer to amino acids in a protein polymer. This change in language is referred to as translation. We'll need to cover a lot more detail than just that, though. Let's begin with the first step, the process of transcription. The process of making an RNA copy takes place in 3 steps: initiation, elongation, and termination. The cell has to determine where and when to start transcribing. It needs a process for transcribing, and it needs to know when it's gone far enough that it has all it needs to make the gene later on. Let's orientate ourselves with how the DNA of a gene presents itself. We call this area the transcription unit. It will have a promoter, an RNA coding sequence, and a terminator. The promoter tells the cell where to begin making an RNA copy. The RNA coding sequence is the code for making protein, and the terminator tells the cell it has gone far enough and to stop transcribing. Recall in DNA synthesis, we used 2 enzymes. DNA polymerase III did most of the synthesis, and DNA polymerase I removes the RNA primers and fills in the rest with the specified nucleotides. In transcription, one polymerase is responsible for transcribing the template DNA strand into a copy of RNA. That enzyme is RNA polymerase. RNA polymerase binds to the template strand at the promoter. Think of the promoter as a beacon. When RNA polymerase sees the promoter, it will attach itself there. The oval circles in this sketch represent the polymerase. The promoter region is a recognizable sequence of nucleotides. You don't need to memorize the sequences. The promoter sequences shown are consensus sequences in prokaryotes, which means that they occur across various bacterial species. It's highly conserved. Attached, the polymerase now knows where to begin transcription. and that is initiation. In order for RNA polymerase to read the book of DNA, the DNA must be unwound to gain access to the individual strand. Recall that Ta bonds only have two hydrogen bonds per pairing, and this makes them easier to break than GC bonds. Therefore, the Ta-rich region of the -10 element helps RNA polymerase unwind the DNA. So just coming back here, we see the -10 element has Ta, TAA, T. This unzipping of the DNA allows the RNA polymerase access to a single-stranded template, and we'll later find out that the direction in which it reads is every bit as important as it was for DNA polymerase 3 and 1. Can you recall what direction DNA polymerase read and wrote in? For RNA polymerase, it is the same direction. It reads the template DNA strand 3' to 5' in direction, and the new RNA strand is synthesized in the 5' to 3' direction. The RNA strand made in transcription has a special name. It is called messenger RNA, or mRNA. It's technically going to be even a pre-mRNA, but we'll get into that into a moment here. Checkpoint 1. In a double-stranded molecule of DNA, which pairs of nitrogenous bases are always found across from each other on complementary strands? In this example, we can see the double strand is separated, forming a bubble where the RNA polymerase can copy the DNA into RNA. The strand the RNA uses is called the template strand. We'll start in the 3' end of the template strand, just like if we were reading a book ourselves. We would always start from the left side of the page and move to the right side. For RNA polymerase, they always start on the 3' side and move to the 5' side. And adding base pairs that match the template strand, building a new strand from the 5' end of RNA to the 3' direction. Now the next letter is an A. If this were DNA polymerase 3, we could predict thymine to pair up with the A or the adenine. However, remember that we are making RNA, not DNA. So what base would pair up with adenine? If you guessed uracil, you were correct. There are no thymines in RNA. Instead, RNA uses uracil, and uracil will always base pair with adenine. What happens when the template strand has a thymine? Well, in DNA, thymines match up with adenines. And because RNA still has guanine, cytosine, uracil, and thymines, there's nothing different here. The RNA polymerase is fully capable of adding an adenine to pair up with the thymine. In this fashion, anytime RNA polymerase needs to add an adenine, it will do so. Anytime it needs to add a thymine, it will use uracil instead. So we've reached another A, and what would be the corresponding letter? AU. So can we remember how the RNA polymerase knows how to call it quits? When does it know that enough sequence has been transcribed to have the full gene message? Just as the DNA had a promoter to tell the RNA polymerase where to begin, there is a second signal called a terminator that tells it when to end transcription. These terminators are sequences of DNA that, when transcribed, form RNA that has structure to it. These structures are the result of the mRNA nucleotides pairing up with each other on the same strand. We can see it right here. The most common example is something called a hairpin loop because it takes the shape of a hairpin. This hairpin, hanging off the back side of the polymerase, causes it to stall, and once it stalls, it becomes unstable, and it is no longer as stably attached to the DNA and becomes derailed. We've now made our mRNA strand through the steps of initiation, elongation, and finally termination. The next step in our journey, now that we have a transcribed mRNA, is the RNA sequence needs to be properly prepped before it can be translated. This is called post-transcriptional modification. We've used the analogy of genes being a recipe. Well, if you've ever looked up a recipe online, you'll notice that you get a lot more than just a plain text recipe. You may have various ways to make the recipe or other sections that may not be exactly necessary to making the dish. That's a lot how mRNA is at this step. It's technically called pre-mRNA. So the next step of the cell is to take out the parts it doesn't want to make the protein exactly how it wants it in this particular instance. It's going to add a couple other things to the mRNA as well. One of which is called the 5' methyl G cap. This cap helps to stabilize the DNA and is a signal to let the cell's enzymes know it is ready for the next step of translation. During post-transcriptional modification, the cell also adds a string of adenine nucleotides to the 3' end, called the poly A tail. This also stabilizes the RNA and protects it from degradation. Nucleic acids often degrade from the ends, so this poly A tail will keep the important parts from degrading before being translated. The stuff the cell wants to copy and paste together to create a plain text recipe are called exons. These are the parts of the recipe that will exit from the nucleus and be sent to the ribosome. The introns are the parts that interject themselves in the recipe, and the cell wants to rid itself of. So the cell splices out all of the introns to make an mRNA with only exons. Don't worry about the start and stop codon. That will be important in the translation step later on. So here we have introns that contain special sequences of nucleotides that indicate to the cell that they should be removed. Small nuclear ribonucleoproteins called snRNPs, pronounced SNRPs, are RNA protein complexes that recognize intron sequences, bind to them, bind to each other to form a spliceosome, and cut out the intron so that exons can rejoin. As I was saying earlier, some parts of the recipe we'd find online could be used to modify the final product. So maybe if you're making a soup, you decide you do or do not want to use onion. The cell can splice together the exons in different ways to lead to different products. These different mRNA products are called isoforms. We may have different isoforms for different types of fur. And here we can see just that with rabbits. Unlike exons, introns do not encode information about the product of a gene. They are chopped out of pre-mRNA before it leaves the nucleus. Introns used to be called junk DNA because they were believed to have no purpose. Now scientists know that they play a role in how genes are turned on or off by different triggers. Point 2. The pre-mRNA molecule did not have a methyl G cap added during post-transcriptional modification. What would be the consequence? Remember, during cellular respiration, we needed to keep track of where things took place in the cell. The same is true here. Transcription and post-transcriptional modification both take place in the nucleus. After the pre-mRNA has been converted to mRNA, it is now capable of leaving through the nucleus membrane to complete the final phase, translation. Translation is the final step and the meat and potatoes of what DNA is all about. That is, to be used by the cell as a blueprint to make proteins. Translation uses the information encoded in the mRNA copy to create a protein. It takes place in the cell's ribosomes. Ribosomes are giant complexes made of RNA and protein. And they're made of the building blocks of protein called amino acids. Like nucleic acids, proteins are polymers made up of monomers. Instead of nucleotides, these monomers are amino acids. DNA uses 4 different nucleotides, while proteins have 20 different amino acids to choose from that all have unique structures and properties. They all share some things in common. They have a central carbon in the middle that is attached to an amine group shown to the left of each carbon, and a carboxylic acid group shown to the right of each amino acid's carbon. At the bottom of each central carbon, you will see their variant group that define which amino acid they are. For example, glycine at the very top left is the simplest of all amino acids and simply has a hydrogen as its variant. Recall that nucleotides are linked by their phosphate ribose bonds, while amino acids are linked by their amine carboxylic acid group bonds. In order for translation to occur, the cells need the mRNA recipe, amino acids for the ingredients, and ribosomes that use mRNA to string amino acids together in the order mRNA dictates, or they're the machinery here. So we learned that DNA polymerase can read DNA code and copy it into a new DNA strand for replication. We learned that RNA polymerase can read DNA code and copy it into a new RNA strand for transcription. Ribosomes can read RNA code and copy it into protein. We're going to need to understand how the code is written in order to see how it deciphers it into amino acids. The mRNA code is written in words called codons. These codons are 3 RNA nucleotides long. So if you have 9 nucleotides, there would be a total of 3 codons there and 3 amino acids. To sense the codons' letters, the ribosomes take advantage of the base pairing mechanism, just like DNA polymerase and RNA polymerase do, but with a slight twist. So let's take a look at how they do this. They employ the use of molecules called transfer RNAs, or tRNA for short. They get the name from the fact that they transfer a amino acid to a growing chain, and they're also, you can remember, they're shaped kind of like a T. The tRNA is a short single strand of RNA. Its base pairs are in a sequence such that it matches up and binds back on itself to form the hairpin loop structures that we saw in the terminator sequences. The business ends here are the 3' end of the tRNA, which each tRNA is specifically bound to only one type of the 20 amino acids. So you may have a tRNA that can only bind to glycine, for example. On the other end of that tRNA is what's called the anticodon, which is a 3-letter section of RNA. Take a moment here to reflect. How do you think the ribosome uses this molecule to read the mRNA codons and figure out which amino acid it codes for? Okay, and again, every tRNA molecule has a 3-letter sequence called an anticodon at one end, amino acid at the other end attached to the 3' end of the RNA, and the anticodon will match up with the codon in the mRNA and deliver its specific amino acid to the ribosome. So it takes advantage of this base pairing effect in order to figure out which amino acid to use. It will grab a tRNA, so the ribosome will grab a tRNA and see if its anticodon will base pair with the mRNA codon. If it doesn't work, it will keep trying until it finds a tRNA that matches. Once it does, the ribosome now knows which amino acid to use. That amino acid is conveniently attached to the same tRNA, so it will take the amino acid from the 3' end of the tRNA and then discard the rest of the tRNA and move on to the next codon. What's crucial here is the translation between these two languages is universal amongst living organisms. This table is what is called a codon chart, and you do not have to memorize the whole chart, but know how to utilize it. It lists all possible 3-letter codons and what amino acid the code of that codon codes for. Around the periphery, you will see it says first letter, second letter, and then third letter. So if I were to give you the codon CAU, let's solve for the amino acid. Start with the first letter and find the C in the row. Next, along the top, find the second letter or the A column. Here's the A column. Now find where the C row and the A column meet. In that box, it must either be a histidine or a glutamine. Using the third letter here, okay, we can figure out that if we take the U, it's going to be a histidine, okay? There are 64 different combinations to be made with 4 nucleotides and 3-letter combinations. But wait a minute, there's only 20 amino acids. And for that reason, there's more than one way to code for each amino acid. There's more than one different codon. Glycine, for example, has 4 different codons that will make a glycine. Usually they're pretty similar to each other, so that if there's a mutation in one of these letters, oftentimes it doesn't result in any major significant changes. So if the third codon here was a U, C, A, or G, we would still get a glycine here. Okay, there are also 2 very, very important codons called the stop codon and the start codon and the start codon. And they're highlighted in green here is the start codon. And there's only one. It's AUG. It encodes for methionine. There's 3 different stop codons, UAA, UAG, and UGA. Checkpoint 3. Use the codon chart. Determine the amino acid sequence of the protein that the following mRNA molecule would be translated to. Here is the ribosome, which is the machinery responsible for protein synthesis. They are made of the small ribosomal subunit and the large ribosomal subunit, and they join together during translation. The two ribosomal subunits clasp an mRNA transcript as they are translating it into protein. Translation, like transcription, has three stages: initiation, elongation, and termination. Here is an example of mRNA ready to be read by the ribosomes. Every mRNA begins with a segment that is not translated into protein. The ribosome has to find the beginning of the protein, of the code for the protein. The signal to begin translation is called the start codon. If you refer to your codon chart or remember, you will see that this is AUG. So here's the untranslated region. Here's our start codon. And the small ribosomal subunit binds to the beginning of the coding region. An initiator tRNA approaches the mRNA. This tRNA has the anticodon UAC and the amino acid methionine. Then the large ribosomal subunit joins the small subunit to clasp the mRNA. The initiator tRNA enters what's called the P site, or the large ribosomal subunit. The A site is where tRNA molecules will flow in and out of. Their anticodons are checked against the codon of the mRNA. If there is a match, then they become bonded to the amino acid in the P site and then fed or passed P site. We have it. The anticodon matches the mRNA codon, so a new amine to carboxylic acid bond forms between methionine and glycine. Then glycine will be passed to the P site, squeezing the methionine out, and the ribosome moves down the mRNA to the next codon. And the new tRNA that has a matching anticodon latches onto the mRNA and binds to glycine. And the process repeats. Notice how the tRNAs now leave and have lost their amino acids in the process, and leaving behind a growing chain of amino acids. So far, we have shown that polymerases read DNA in a 3' to 5' fashion. But just be aware that the ribosome reads the mRNA in a 5' to 3' fashion. When the ribosome reaches a stop codon, a release factor approaches, which is a protein that stops translation. The release factor signals for the ribosome to fall apart, and now translation of the protein is complete. So this release factor is going to bind to the stop codon. And here is the final string of amino acids derived from the codons of the mRNA. However, this is far from the end of the formation of a protein. Proteins, just like the ribosomes, are complex, three-dimensional, and dynamic structures. And we now need to learn just how we go from a strand of amino acids to these complex tools of the cell. We're now going to watch a video that puts all of this together. So right off the bat in this image, we can see that there's the chromosome, and the animation has depicted a less condensed form of chromatin here. Here's the histones, which help package the DNA tightly. When the histones are not winding up the DNA, then we get exposure of our double helix. Okay, so here we have a highlighted gene here. Okay, so the RNA polymerase has attached to the start of the gene. Remember, it does so through the action of a promoter. And the promoter region, such as the -10 element, is rich in A's and T's, which allows the DNA to be separated by the RNA polymerase more easily. And now it's going to start, since it's initiated, it will start the elongation. And we in our drawing so far, we've just had a single nucleotide just appear out of nowhere. But really, what's going on in the nucleus is there's just millions of RNA molecules floating around. And so it's by chance that these RNA molecules come into close proximity. And if they match, they're going to bind up with the DNA strand. And then the RNA polymerase's job is to connect them through their ribose phosphate bonds to each other. So here we go. And remember, it's this bottom template strand is being read from the 3' direction to the 5' direction. And the new code that's being written is 5' to 3'. And here's our fresh messenger RNA. It's actually a pre-mRNA, mRNA. And so it spliced out the introns and stuck the exons together using the spliceosome. And now that it's prepared as a mature mRNA and it has that 5' cap and the poly A tail, it's able to go through these nucleopores of the nucleus into the cytoplasm. And here's the ribosome, the large and the small subunit. The small subunit is supposed to bind first. It's attached here, um, and it begins synthesizing the new protein at the start codon, which is an AUG. And here's our tRNA. We have our anticodon at the bottom, the amino acid attached to the top. As you will recall, AUG or methionine is going to be the very first amino acid attached at the start codon, which acts as the signal. And there's the anticodon. Notice the tRNA is falling off without the amino acid. Yes, and there's the growing chain of amino acids bound via their amine and carboxylic acid groups. They come together to form the chain. Now it's just a strand. It's just translating the language of nucleic acids into the language of amino acids. The last amino acid is determined based upon the stop codon presence. Yes, and now that single strand has to fold upon itself in order to form this complex protein molecule. And if you remember back into an earlier chapter, we discussed part of how this is accomplished is The cytoplasm is an aqueous solution, or it's a solution with water as the solvent, and that's a polar solution. And so the nonpolar amino acids will want to shield themselves towards the inside, and the polar amino acids will want to face outwards towards the solution. This is a very similar mechanism to how lipid membranes form. Remember, the phosphate group on a lipid membrane points towards the solution of water, either inside the cell or outside. And the lipid tails, the fatty acid tails, face inward away from the water because they are nonpolar and the phosphate is polar. And there we have it. Every amino acid has different chemical properties based on its variant group, sometimes called its R group, which is highlighted in this chart. These chemical properties will define how a protein folds, and how it folds will define how it functions. So how does changing the DNA change the protein? Examine the following mutation. Here's the original strand, which has a G that has been substituted out for a C, and a mutant strand. So what effect will this mutation have on the amino acid sequence of the resulting protein? Well, let's divide it up into its respective codons. Okay, and then we can see we have a completely different codon, and the anticodon of this may introduce a different protein. Using the codon chart, let's find the original CAG. The first letter is a C, so we start on the second row. The second letter is an A, so we take the third column. Okay, and the letter G tells us that a glutamine is the amino acid encoded by CAG. However, the mutation changed the CAG to a CAC, and so instead of a glutamine, we get a histidine. Something important to note here. Look again at the histidine glutamine box, and what would happen if instead of a CAC, the mutation was a CAA? Well, ultimately, we would still get a glutamine. This is called a silent mutation, because the mutation did not lead to a change in the amino acid sequence. But we did not get a silent mutation. We got something else called a missense mutation. And so our mutant would have one change in the amino acid structure. When a mutation in the nucleotide sequence leads to a change of one amino acid to another, this is called a missense, like so. Tech point 4. Using the codon table, provide an example of a single nucleic acid mutation that would be considered a missense mutation. Okay, how does changing the DNA change the protein? So let's do another example. Here we have the original code and a mutation from a U to a C. And what effect will this mutation have on the amino acid sequence of the resulting protein? So UAU is tyrosine, and UAC is also tyrosine. And so therefore, this mutation will lead to no changes. And this is because multiple codons can translate into the same amino acid. There are 64 different codons, but only 20 different amino acids. Therefore, there is more than one codon per amino acid. Because of this, some mutations have no effect on amino acid sequence, and such mutations are called silent mutations. Checkpoint 5. Using the codon table, provide an example of a mutation that would be considered a silent mutation. Here is another example. Again, we have the original DNA sequence and the mutant sequence. We'll divide it up into the codon structure, and then we want to compare the UGC codon to the UGA codon. So the UGC codon is going to be cysteine, and the UGA codon is going to be a stop codon. So let's write out the protein sequence now, and we see that what we have done is something even more dramatic than a missense. A mutational change where a normal codon is turned into a stop codon is called a nonsense mutation. The three stop codons do not encode an amino acid, but rather indicate that translation should stop at this point. A mutation that changes a normal codon into a stop codon is called a nonsense mutation. Truncating the protein can have massively detrimental effects to the function of a protein. Sometimes a mutation occurs where an extra nucleotide is added by mistake. What effect will this mutation have on the amino acid sequence of the resulting protein? So we'll break it up into the codons, and here we can see something interesting. This will shift all of the amino acids down by one. Well, all of the nucleotides down by one. So all of the codons will be shifted. And if we jump to the conclusion here, if we were to use the codon chart, we can see that the first amino acid would be the same, but every subsequent amino acid would be different. And so this is going to be read completely differently by the ribosome. This is called a frame shift mutation, which results when one or more nucleotides is inserted or deleted. This causes the reading frame of the DNA codons to shift, impacting the translation of all downstream codons. Since codons are read as three-letter words, we can see how dramatic the effects are by taking the sentence, "The dog ran for his log." They're all three-letter words, and how the addition of the letter B makes the sentence a complete jumble. There are two ways to get mutations. One are inherited mutations, where mutated alleles are inherited from an organism's parents. Inherited mutations affect all cells in the body because they are passed down through the sperm and or egg, which form the zygote. Acquired mutations are mutations that arise in individual cells in the body and then spread as those cells multiply. These mutations are acquired sometime throughout a person's life due to random errors or exposure to mutagens. And when we say random errors, we're talking about when the cell repairs or replicates its DNA. Sickle cell anemia is an example of an inherited mutation in the protein called hemoglobin. This protein is responsible for carrying oxygen through the circulatory system to the body's varying tissues. This is the sequence for normal hemoglobin subunit. And the sickle cell allele has a substitution of an A for a T right here. So here's the ATG or AUG if in mRNA. That's the start codon. So not too far down the road here, we have a substitution for the what was an A for a T. And when we use the codon chart, here we have this first 7 codons of the allele for normal hemoglobin. And here is the codon chart for the following checkpoint question. So I gave you the 7 codons, amino acids for the normal hemoglobin protein. For the sickle cell allele, what amino acids will it be translated into? For the allele for abnormal hemoglobin, the following. These two proteins of hemoglobin are dramatically different in their characteristics. Valine is nonpolar and hydrophobic, while glutamic acid is electrically charged. So what impact will this have on such a crucial protein? This one amino acid change has an incredible impact on the shape of the protein and leads to the hemoglobin clumping together, causing the shape of the red blood cell to form a sickle cell shape, which can interfere with the blood flow. Checkpoint 7. Using the codon translation table and your knowledge of amino acid chemical properties, which of the following mutations do you predict would have a greater impact on an organism and why? All right, so that was just an example of an inherited mutation. Let's go ahead and look at one that arises through an acquired one. So sickle cell, again, it's a hereditary mutation. And an in this case study of the RB1 tumor suppressor is going to be one of an acquired mutation. And these mutations can arise in individual cells as a result of DNA replication mistakes or environmental mutagen exposure. And then when these mutations affect a gene that is important for cell cycle regulation or tumor suppression, the result is cancer. So in this case study, Mary is a 42-year-old mother of three. She has smoked cigarettes since she was 19. Mary developed a persistent cough some time ago. In the last few days, she has developed tremors in her hands and other neurological symptoms. Here's an x ray of her lungs 2 years prior, perfectly healthy and clear. And currently, there is a tumor present in the lungs. A biopsy of that region of the lungs reveals squamous cell carcinoma from a lung. Squamous cell are when the cells enlarge and take on sort of a squarish shape to them. And this is an indication that those cells are cancer cells. And what happened was the cancer cells metastasized by moving into the bloodstream or lymph nodes, which carry the mutant cells to other parts of the body. And a metastatic tumor was the cause of Mary's hand tremors and neurological symptoms. Every cancer is caused by a different set of mutations. In Mary's case, genetic analysis revealed mutation in the gene for retinoblastoma 1, RB1, a tumor suppressor protein. And this tumor suppressor protein, when it's functioning properly, is normally involved in the G1 checkpoint of the cell cycle. Remember, this checkpoint, everything, the cell has to be operating normally in order for it to continue with the S phase where it replicates its DNA. And if this retinoblastoma 1 is not working properly, cells that should not be going into S phase are going into S phase. And this leads to a somewhat uncontrolled growth. And that's kind of what cancer is. A normal RB1 gene is almost 5,000 nucleotides long. In this segment, the bottom line begins at the 2,641st nucleotide. And a few nucleotides down the way here, when the tumor cells had their DNA sequenced, a deletion mutation was found in this area of the gene. How would Mary's mutation be classified? Checkpoint 8. Various compounds in the cigarette damaged the DNA in one or more of Mary's lung cells at this location in the RB1 gene. And a substance that mutates DNA are called mutagens. Those that mutate DNA in a way that cause cancer are called carcinogens. Sunlight, specifically ultraviolet radiation, can also lead to mutations. Sunlight causes thymines found side by side on the same strand to bind to one another. This is not normal. Usually they're just bound through the backbone of the phosphate ribose. But here, the individual carbons of the nitrogenous base are binding to each other, and it's called a thymine dimer, and it causes DNA polymerase to incorrectly read the DNA during replication or repair. And so what happens is the wrong base pairs get put in here during repair or replication. Okay, and that is the end of chapter 15. Thank you for staying through the whole video, and please join us for the next one.